You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you. Hello, this is Richard Jacobs with the Future Tech and Finding Genius podcast. I have Joseph Vallino. He's a senior scientist at MBL, which is Marine Biology Labs. We're going to be talking about the various work that they're doing. So, Joseph, thanks for coming. How are you doing?
I'm doing good. Thanks for having me.
Yeah, if you would, tell me about your research, because it's pretty descriptive here, but I don't want to mess it up. I'd rather it come from you where it's clear as to what you do.
All right. My research focuses mostly on marine chemistry, in particular marine biogeochemistry, which is basically the study of how mostly microscopic organisms, primarily bacteria and other plankton in the ocean, modify the chemistry of the ocean in terms of nutrient cycling, such as consuming things like nitrate and fertilizer, nitrate and phosphate, which are more or less like fertilizer in the ocean, using solar radiation to drive biosynthesis. And when you combine all of these microbial processes that occur in the ocean, you get the overall biogeochemistry that's occurring. So in terms of things like fixing carbon dioxide out of the atmosphere, producing oxygen, cycling nitrogen, cycling phosphorus, and cycling many other elements, is largely governed by microbes then. And similar types of processes, which I also study, but not as much, also occur in terrestrial systems, largely in soils or in sediments, or in sediments in the ocean. And trying to understand how those microorganisms function, in particular, the kind of chemistry they conduct, is the primary area of research. And I'm mostly using mathematical models to understand that. So that's
who are the biggest players in the ocean, and what are they doing? Like, what are some of their jobs?
Well, in terms of, the classic examples would be the phytoplankton, so things like diatoms and dinoflagellates that are most microscopic organisms that are using solar radiation to fix CO2. So they're the base of the food web. So everything you see in the ocean, from small crustaceans all the way up to whales, are reliant then on that primary production that's orchestrated by those phytoplankton. So this is different than what you find on land, where the primary productivity is largely by trees and grasses, by very large macroorganisms that are the primary producers, the primary base of the food web. In the ocean, it's all, except for in coastal areas where you have things like kelp and other large macrophytes, by and large, most of the productivity in the ocean is by these microscopic organisms.
So how is it different then if a microscopic organism is fixing carbon? Is it fundamentally different, the process or the efficiency or other aspects of it, than when large organisms like trees and grasses do it?
By and large, no. I mean, there's some subtle differences in terms of the type of metabolism, whether it's what's called C3 or C4 or some other types of way of fixing carbon dioxide. But in general, the basic principle is the same. The difference is kind of what's kind of constraints on those systems. And this is kind of where the fundamental aspects of my research come into play. And the reason is there's a challenge when trying to understand all these microscopic organisms in terms of how active they are and what they're doing. So in the ocean, as well as other environments, as I mentioned, there are, for instance, in the ocean, there's 1 billion bacteria per liter of water. And in that liter of water, there might be upwards of 1,000 or even 10,000 different so-called species of bacteria that can be found in that water column. And trying to understand then, that number of organisms becomes very challenging. And so I've been developing some different approaches based on ideas that derive from thermodynamics to try to describe the overall chemistry that sets up without necessarily knowing exactly what players are there, because it's very difficult to predict who's there and how active they are at any instance in time. And so
instead of looking at all the species, what are the main jobs that a section of water accomplishes? So it's the carbon fixation, any other major activities that are going on there.
Yes, and so that it's that functional aspect that really is the focus. So you have carbon dioxide fixation, you have just kind of standard respiration like we do. You're taking organic materials, you're taking in food and you're oxidizing it. In areas of, there's also quite a bit of nitrogen fixation, so that when the ocean or the surface ocean in particular becomes limited by nitrogen, there are plankton and bacteria that can fix nitrogen out of the atmosphere. There are places in the ocean where you have what are referred to as oxygen minimum zones, where oxygen is near becoming unavailable because it's being drawn down. And in these cases, you have bacteria that can use sulfate, so they can breathe with sulfate instead of oxygen, or they can use nitrate instead of oxygen. So there's quite a bit of other metabolic pathways that only bacteria can catalyze that allow these systems then to cycle nitrogen and sulfur and other elements that are essential for life. So those diversity of metabolic processes then is usually the focus of what I'm trying to look at. So you can kind of look at the overall chemistry being set up in the ocean as a big metabolic network. However, that network is distributed in the sense there's different organisms that are contributing different metabolic functions. So although the metabolism in some regards is similar to what you might find in kind of an abstract way, what a single cell might be carrying out. In this case, it's not a single organism, but rather this distributed nature of the system. And the challenge then becomes, how do you describe this overall metabolism of the ocean when it's distributed? It's not under any centralized control. And so some of these ideas then is, in terms of thermodynamic ideas, are this idea that systems organize to dissipate energy. So when systems have many different ways of assembling, they have many moving parts, so to speak. Conjecture is that systems will organize in such a way as to maximize the dissipation of energy. An example
living
of that,
systems or non-living systems, I would think that they act very differently.
Right. So that's where it starts to become interesting. So the theories which go under the name of maximum entropy production aren't they're general theories, and they apply to both non-living as well as living systems. So an example of a non-living system would be the formation of a hurricane. So a hurricane is a nice example of an ordered structure that organizes, and its presence actually increases the rate of heat transport from the ocean to the atmosphere that's built up over the summer. So you have this solar radiation that's heating up the ocean over the course of the summer. You're building up this large thermal gradient between the ocean and the atmosphere. And if the system can organize to increase that dissipation of that gradient, that thermal gradient, then it's expected, or there's a higher probability of such a system organizing. And so that's the basis of a hurricane. The extension I'm applying is taking that same kind of idea and applying it to biological systems. And that is, if there is some energy available, some food available in the system, such as there's glucose and there's oxygen present in the environment, that the system will organize in such a way as to try to destroy that chemical potential, to oxidize that glucose, that food, into CO2 and water. And that's then a very basic principle that I'm exploring in terms of trying to describe how then living organisms organize. And so the general idea is to kind of describe the overall metabolism of, for instance, the ocean as being composed of all these different organisms. And those organisms that will dominate at a particular location and at a particular time are the ones that are able to dissipate energy most effectively. So it kind of represents.
Okay.
Yeah,
living systems in the ocean. You said they're distributed. Where are they distributed? To give people an idea that don't know, what happens at the surface of a particular body of water, and how far down does the biological activity go, and at what points does it change? And just give me an idea spatially.
Yeah. So in one regards, the microbes are distributed pretty much everywhere. They can, as I mentioned, there's a large, there's 1 billion bacteria per liter of water. Most of those bacteria, though, will be at low numbers. There won't be that many of them. They'll only become dominant where their niche, their expertise, so to speak, matches the niche. So there's areas where there's, for instance, lots of nitrogen, lots of fertilizer available, and organisms that can utilize nutrients at high concentration will dominate. Other areas of the ocean, like in the middle of the oceans and the ocean gyres, where the nutrients are extremely low concentrations, you'll have different species that are doing the same thing in terms of fixing carbon dioxide and chewing up organic matter that's produced by the primary producers, but they're able to utilize nutrients at extremely low concentrations. You'll have other organisms that specialize at operating at relatively higher temperatures, and other ones that operate at low temperatures. Ones that operate at high light levels, other ones that operate at low levels. But in terms of the biological, where most of the biology is occurring, is in the top 100 meters of the water of the ocean. But it can be as you get into areas that are more so-called deserts of the ocean, the middle of the oceans, the gyres, you can the depth of the active organisms can go down to, in terms of the photosynthetic organisms, down to maybe 1,000 meters, a kilometer. But microorganisms are then active, of course, throughout the whole water column, from the surface all the way down to the abyssal plains of the ocean and deeper into the trenches. And then you get into even more interesting aspects when you get into hydrothermal vents and the microorganisms that are living at the bottom of the ocean, where vent waters are present.
If you compare, let's say I'm at a beach, and what does the water composition and the microbial composition and activity look like, 100 feet out in the water where the waves are active versus right near the shore? Has that been looked at? Or if there's an algal bloom, have you compared, again, what's going on locally, at least, to get a sense of how much things change and how they are in various locales?
Right. That's mostly driven by, again, kind of nutrient availability. So in terms of the, energy coming in from the sun, that's going to be the same whether you're in the middle of the ocean or on the coast. But when you're closer to the coast, you have, first of all, lots of nutrients that are, leaving the terrestrial land, or the land, and being exported to the coastal zone by rivers. And that river water carries a lot of nutrients. Even under natural conditions, there's a fair amount of nutrients in water and fresh water. But of course, as we develop the coastal zone, there's even more nutrients available. So in general, you'll have much higher primary productivity, so much higher rates of carbon dioxide fixation and oxygen, and just basically much higher cycling rates occurring as you're closer to the coast, within a kilometer or so of the coast. In other areas, also along the coast, there can be what's called upwelling of deep water that also is high in nutrients. So in general, those two kind of processes govern the amount of nutrients that are being exported in the coastal zone. So you'll have very high productivity there. You'll have lots of support. That high productivity will support fisheries and other higher organisms at a much higher rate. But then as you go out towards, again, more the central gyres of the oceans, you'll start to effectively deplete those nutrients. And so the concentration of nutrients will be significantly less, maybe 100-fold less compared to the coastal zone. And so you'll have these those gradients. So people do, as you mentioned, study the transects from going close to shore to offshore, and looking at both the nutrients availability and the organisms that are associated with utilizing those nutrients and how they change over those scales.
Have you studied the garbage patches where supposedly there's a mile of microplastics and plastics concentrated in a gyre? Have you gone out to those locations to look at the microbial activity?
No, so I mostly focus in the coastal zone, mostly in estuarine work. So the estuary being that kind of interface between freshwater and full marine water. So in those areas where there's large or small rivers entering the oceans, those are these estuarine environments. So I predominantly focus in those coastal areas, and not.
So what is the so what's the specific focus of your research? What are you trying to understand specifically and modulate or know about the regions you study?
Right. So it's at this point, it's mostly trying to develop predictive computer models. So if you wanted to, as you have a weather model and you can try to forecast what the weather is going to be like tomorrow or a week from tomorrow, we can develop similar models that operate within the ocean and try to predict how nutrients coming from land or just being cycled locally fund or fuel that primary productivity, that growth of the phytoplankton. And that's where some of the challenge lies, in the sense, as I mentioned before, that you have such a large number of bacteria and phytoplankton in different species. It's no longer practical to try to model and describe the interaction of all these organisms, of these thousands and thousands of organisms. And so what I've been trying to do is to test this hypothesis of trying to describe, at least at this kind of very high level, at this functional level, that is in terms of CO2 fixation and nitrogen fixation and sulfur cycling, can those processes that are governed by biology be described by this rather simple thermodynamic principle, this maximum entropy production idea? And so it largely comes down to first developing the governing equations that describe those systems, and then seeing how well by a combination of either experiments and or field work, going out and sampling, and seeing whether or not those models are able to predict what you observe. It'd be similar in the weather. If you're in early days of weather modeling, you would develop a model to describe the weather, and you would then have some forecast of what's going to happen tomorrow and the next day, and you could see whether or not your forecast matched the observation.
What about looking at it in a slightly easier way? Is look at one, like if you look at the things that go on typically as jobs to be done. I don't know who the taskmaster is, but these jobs need to be done or are done. Nitrogen fixation, CO2 fixation, etc. If you look at just one of the jobs and the players that accomplish that job, maybe that's a way to look at this in a more narrow way that would help you understand, at least within the context of that one thing going on, what's driving it, what are the dynamics of it.
Right. So I have done those kinds of experiments over the last decade or so, and it's kind of moving into the field now at this point. But that is the approach taken. So there is an aspect of trying to model this community of microorganisms. So studying one organism, you can do that fairly well. But the problem is when you go out into either an experimental system I set up in the lab, or I go out into the field and you look at, for instance, let's say that the main players that are fixing nitrogen or fixing CO2 or utilizing methane if it's present, what you often find is that the main players constantly change over. So that if I go out there today and I'm using molecular techniques, or I work with colleagues that can employ those techniques, and we can look at and see what the dominant species are that are fixing nitrogen. I can then come back the next day or a week from then, and there's a different organism there that's doing that. And what you actually often find is that there's this continuous turnover of microorganisms that are carrying out that function. It would be as if you went to a forest, and there are pine trees in this forest one day, and you come back and the next, and there's birch trees, and then there's a whole succession. And that does occur, but in terrestrial systems, it takes thousands of years to see that kind of cascade of different players occupying the primary spots. In the microbial world, that happens really fast. It can happen in the order of hours or days, and you have this kind of continuous replacement.
Yeah, I guess, again, there's a job to be done and there's redundancy. Different players can do the job. So I guess because of the mixing and moving and, the non-still nature of bodies of water, that those things turn over a lot. Have you tried to compare it to a lake or a still body of water to see if
so it's
a lot less difference.
Yeah, so it's not that large scale mixing that drives it. So I do run these in 1 liter or 10 liter microcosms, and you can see that same changing of the community. So what appears to be happening, or one hypothesis, is that as the community starts to grow up, it slightly changes its environment, or radically changes the environment. It'll change the pH. It'll use up all one compound, or it'll produce another compound. And as that local environment, that might be local being a centimeter or some small volume, as those conditions change, it gives rise to a potentially new niche. And so some other organism that's present that can take advantage of those slightly different conditions starts to dominate. So as there's this kind of continuous niche selection, as the organisms constantly modify their environment, they then give rise to this kind of succession. So although...
What's interesting, so when you say, when one is predominant, in terms of numbers, I don't know if you could do that, ballpark numbers, what would happen in a small volume of liquid? Like you see strain A predominate and then strain B and then C, and how much are their numbers changing? And when the strain now wanes, what happens to all the bacteria that compose it? Are they dying and being eaten by other bacteria, or what's happening?
Those are all good questions. But in terms of the numbers, for instance, when we ran a microcosm experiment a few years back, looking at this question, we could find that the dominant, in this case, it was we were bubbling it with methane, and so it turns out there are methanotrophs that can take methane and burn it with oxygen. And those methanotrophs, at anytime we sampled it, there might be a single methanotroph that was 60 to up to 85% of the population. So almost all the population is dominated by this single species of bacteria, methanotroph. If we looked a week later, we might find that there is some other species now occupying 60 to 80% of the population. And the one that we measured the previous week, its value might be below the level of detection. In this case, the level of detection was about 0.01%. So we have something that goes from less than 0.01% up to 85% of the population, and then back down to 0.01% in the course of, I'm not sure how. It's certainly less than a week. I think the quickest we sampled it was over a couple of days, and we were still seeing those wild fluctuations. So you can go from what's referred to as the rare biosphere, that is, you go out and sample the environment, and although it's dominated by a few bacteria, you'll find a huge number that are at very, very low abundances. And these organisms at low abundance are members of this so-called rare biosphere. And it appears, at least under certain situations, those members can become the dominant members for a short period of time and then be returned to that rare biosphere. Now, what's driving that is not, is it, mortality by other grazers? There's, of course, protists that will graze bacteria. There's viruses that are causing viral, bacterial lysis. And there's simply just being, lost because you're being evicted out. Water movement is taking you out of an area. So those are areas of active research in terms of trying to determine what drives those internal dynamics.
Well, when you see a change from A to B, I would think the metabolite profile would change dramatically, the concentration in that little bit of water.
Yeah,
and then also what happens again to the corpses of the corpses of all the bacteria that are no longer present. Where do they go? Who eats them? What happens to them?
Yeah, so they're being they have to be being removed. So you're correct in that regard. Whether it's by viral lysis or bacteria, bacteria also eat bacteria. There's bacteria predators, and like I said, organisms that are slightly larger, that are eukaryotic protists that will eat them. They're turning the bacteria are getting turned over very quickly in the ocean. Once per day. So there's a very high mortality rate. So there's a high growth rate. The bacteria in the ocean like I said, will turn over about once per day. Now that's, to compare it to a laboratory, if you're growing E. coli, they can grow as fast as turning over 50 per day. And that's a combination of, again, persistent grazing and viruses. So there's this high growth rate, a relatively high growth rate, and a high predation rate. So it's a very fast wheel that's spinning. And so, like I said, as these niches open up, then other organisms can take their place. In terms of the metabolites, the exometabolite pool or exometabolome, that's also an area of active research. And my group's just starting to kind of explore that, or working with colleagues that are exploring that, so that you can characterize, well, how is that how's that microenvironment changing over time? So that's again an area of active research.
Do we have the tools to be able to do microscopy, without disturbing the sample, to literally watch what's going on as it's happening in changeover?
Most of it, in terms of the microscopy, I would say it's probably too dilute to a certain extent. Even though I said there's a billion bacteria per liter, it turns out that that's not very high density. You can get, in a biofilm, you can get up many more orders of magnitude than that. So microscopy is not usually the tool of study. It's mostly driven by a combination of molecular tools, so looking at metatranscriptomics and metagenomics. So looking at the DNA and RNA that's present in the sample, and looking at how the relative abundances of either genes that target what species, what bacterial species are present, or what functional genes are present. For instance, genes associated with CO2 fixation. And so those are one set of tools. The other sets of tools are metabolomics, or mostly exometabolomics. So it's looking at metabolites that are outside the cell. Another area active area that's being explored is proteomics, so looking at what proteins are present. So in terms of difficulty, the metagenomics and metatranscriptomics are fairly well developed nowadays, and people apply those routinely. Metabolomics and proteomics, those are also being applied, but in natural waters, concentrations are very low. So people do it, but it requires a lot of expertise and some specialized equipment to do that. But that's definitely where the field is heading, is trying to tease apart the underlying genetics, as well as the machinery in terms of protein and metabolites that are being produced and consumed in the environment.
Well, what if you did a biomass balance? If you took just like a milliliter, of seawater, or maybe even a liter, and you let it sit for a while, and it undergoes change, and you were able to somehow estimate the amount of biomass in it, does that not change at all? The corpses are being eaten, or does it grow or shrink?
It doesn't change that much. So if it's certainly like a natural system. So if we go out and sample the ocean, I'll bring back the million bacteria per milliliter, or billion bacteria per liter. That number doesn't change very much. It'll maybe bounce around, by a factor of 10. But in general, there's if I go out into the coast right now, or out in the middle of the ocean, it might be a little bit less. But the rule of thumb, you'll find it is a million bacteria per mil, and it doesn't change that much in terms of that biomass. So you're not seeing big swings in total biomass. It's relatively constant. It's quite surprising just how constant, or relatively constant, that value is.
Well, that's a good clue, because again, if A dominates and then B dominates later, does that mean that B is eating A? Otherwise, the biomass would increase, or is that biomass of A being distributed amongst all the other players, and so their percentages would change?
Yeah, so it's more as if their resources are being distributed. So if you have bacteria A that's dominating the system, and you come back some short time later and it's bacteria B that's dominating. Bacteria A was basically grazed by a combination of predation, like I said, by bacteria, by viruses, by protists, and then of course there's a food web, and so those organisms, those protists in particular, that are eating the bacteria, there's other ones that are eating those, and what occurs is a very kind of tight tight recycling loop, so that the carbon and the nitrogen and all their elements that were locked up in bacteria A ultimately get recycled into the environment. And as long as there's energy being put into the system by, for instance, by solar radiation, those now resources that are back in the environment will then be utilized by another set of bacteria and other plankton to consume those resources that are being released. So it's a very tight spinning cycle, if you will, that's made up of many different parts and continues to function in this kind of homeostatic way, even though the individual parts, though, are quite dynamic. And this is why it becomes quite the challenge to try to understand it and try to come up with a model that can represent that. And so the idea is that
it'll be interesting to see how many creatures does you had bacteria A dominates and then it wanes. How many different creatures take up the material, and how many creatures does it cycle through before it comes back to A, let's say, or how many hands does it pass through, I guess you could say.
Yeah, so we can study that to some degree, and actually I have an experiment going on right now looking at some aspects of that. So this is actually, again, a laboratory experiment where we have basically what's called a chemostat, which is basically a bucket where you have media coming in and then the contents of the bucket emptying out, so that the volume of the bucket is constant, yet you're constantly putting in new materials, constantly putting in fresh media. And the bioreactor in the bucket is basically just a natural microbial community. We quite literally just went to a pond and collected some water. So it has a full diversity of microorganisms present. We add it to the bioreactor, and we basically supply a feed of media. But what we can do is in that media, which is now a defined media that has things like glucose and ethanol and methanol and a few other metabolites, a couple other food sources, we can actually label one of the carbon sources, for instance, like glucose, and we can change it from C12, which is the most abundant form of carbon, to C13, which is a stable version, a stable isotope of carbon, but it's heavy. Now, as the bacteria consume that heavy isotope, if we at a point in time we change the glucose from C12, the normal glucose, to C13 glucose, the heavy version of glucose, the bacteria will consume that glucose because they can't tell the difference. And as they build their biomass, as they build DNA and RNA and protein, they'll incorporate that heavy isotope of carbon into their biomass. You can now take a sample from the bioreactor and put it into an ultracentrifuge, and that ultracentrifuge can actually separate the DNA or the RNA based on the different weights of C12 versus C13. And so you'll end up, if you can picture a tube that's been put into an ultracentrifuge, and there'll be two bands, kind of like a layer cake you might imagine, and the top band will be composed of DNA or RNA that's based on C12, and then the heavier band that'll be below that C12 will be the bacteria that were only taking up that heavy glucose that was labeled at the C13, but also the organisms that were consuming the bacteria, so the protists as well as the viruses, will also start to pick up that heavy isotope, and you can now go out and sequence those two bands, and you can see who wasn't taking up the glucose because they're in the C12 band, and you can see which organisms were directly taking up the glucose, and then the organisms that are eating those organisms. So you can start to reconstruct, to a certain extent, who is eating what, and then to a certain extent, the organisms that are eating those organisms. So you can start to reconstruct the food web using those kinds of tracer techniques.
Cool. Very cool. So what do you feel like you're on the verge of making a breakthrough in understanding, maybe in the next year or so? Anything in the near term?
Yes, the breakthrough is always a kind of a fun question, or that, in terms of my line of research, it's I don't really necessarily expect a breakthrough in the context that you often think of, in the sense that, if you're experimentally driven, you usually have some objective that you're trying to achieve. And when you achieve that objective, you kind of have a breakthrough. In terms of the kind of the mathematical models I develop, it's more of a kind of an iteration where you're trying to improve the models, and at the same token, you're trying to convince people that, largely other scientists, that this idea that systems organize to dissipate energy is a viable way of describing biology. Because it's a little bit counter to the standard dogma in biology. Most people studying biology are thinking about individual organisms and how those organisms grow. Under what conditions did they grow, what food are they eating, and things of that nature. And that's very important research. But this is taking it from a different perspective. It's a colleague of mine, Charlie Lyman, had a quote of it's a paradigm shift from we eat food to food has produced us to eat it. And that is, you're putting the driver on this energy gradient. It's really this energy gradient in the form of, whether it's light or it's glucose and oxygen, that's the driver. And then the system organizes, in this case, what is now what we consider life, organizes to dissipate that gradient. So no one's going to, I'm not going to, it's very difficult, if not impossible, to have a single experiment that will show that that principle is valid. Instead, it's more or less showing that this hypothesis does explain different systems. And over time, and as more people start to explore it, there will be, it'll work. That is, this idea is correct, or no, it's not very good. There's too many caveats, there's too many exceptions to make it a useful idea. So it's more of a building trajectory than it is necessarily a breakthrough discovery, as boring as that sounds.
Well, what's your, do you believe that living systems and non-living systems are fundamentally different? Or do you think that they're the same? We just, we haven't uncovered the principles that guide both.
They're the same in this context. And the principle, or one of the principles, is this aspect that systems organize to dissipate energy. Now that's, that's not going to be, again, we're trying to come up with some simple rules that really explain it. So right now, there aren't any rules, or, if you build a model, there's no fundamental rule that's in your model. You basically put them all together. If you're looking at microbial communities like I do, whether you're studying the environment or you're studying the human microbiome, which is similar complexities, you have basically a bunch of organisms. You take a reductionist perspective and you try to describe how each organism grows, what it likes to eat, what inhibits it, and then you put hundreds of these organisms in together, and it turns out these models then have hundreds of parameters that govern all these nuances of how organisms grow. We don't have a very good handle on any of these parameters, and so it turns out you have a model that you have many knobs you can dial in and tune, and so you can get that model to fit the observations. The problem is usually when you take that same model that you've now tuned to some experimental set of observations or some real observations in the environment, and you run it under slightly different conditions, you usually find that the model, completely breaks. It predicts the completely wrong things, or it doesn't match the observations at all. And that's because there's nothing fundamental in the model that's governing its organization, its structure. You just have a bunch of what effectively comes down to a bunch of big nonlinear model with lots of parameters. If we have an idea that, this principle that systems organize to dissipate energy, that principle, if it's correct, will operate in the future or under different conditions that we haven't examined a system under. And so you can use that as a way to try to describe how systems organize. So whether or not it's, life on this planet or life on Europa, or the idea is that if systems can be described by this, you have at least some predictive capability that you can try to develop a model around.
Well, very good. What's the best way for people to find out more about your research and to, read papers, get more information?
Well, there's my website, eco37.mbl.edu would be at least somewhere some of my publications are at, and within those publications are more. In terms of general websites, not too many yet in terms of, as this kind of idea is being explored. Again, its success will be more or less measured by its popularity in terms of other people, utilizing it to try to describe how living systems organize.
Right. Well, very good. Well, Joseph, thank you for coming. I appreciate it. It's really interesting stuff you're working on.
Well, thank you, and thanks for chatting.
You're listening to the Future Tech Podcast with Richard Jacobs. Future technologies such as artificial intelligence, stem cells, 3D printing, gene editing, Bitcoin, blockchain, the microbiome, quantum computing, virtual reality, and exploring space are much closer than you might think. In fact, many early versions of these technologies are in play right now, and the companies that are using these technologies are the focus of this podcast. My goal for you, the listener, is to learn from these podcasts. You may very well learn something that may change the course of your life for the better, steer you towards a new career, or give you insight into addressing a thorny medical problem. Remember, this podcast and its content is informational in nature only. No medical, tax, legal, financial, or psychological advice is being given. If you've enjoyed the podcast, please listen, subscribe, like, and tell your friends about it. Thank you.